we're going to pick up with synapses. Okay. And I'm going to minimize this window so it doesn't obscure my view of the PowerPoint. Um, so if somebody hops in to the weight room, I won't necessarily be able to see it. So if someone wouldn't mind just kind of giving me a shout, that would be great. Okay, so synapses. Synapses, as we have discussed, are connections. So synapses, and these connections are the ways in which a neuron crosstalks with another neuron or is crosstalking with a muscle fiber or a gland. So far and away, the most common type of synapse that we see when we look at neurons are what we call chemical synapses. And chemical synapses, these are the kind of synapses that I was discussing yesterday, and the kind of synapse that we saw when we were looking at the neuromuscular junction. What characterizes a chemical synapse is that you have the synaptic knob not making physical contact, but instead hovering right above the target cell with a space between the two, and we call that space the synaptic cleft. And the chemical part of the synapse tells you that neurotransmitters are used for one neuron to communicate again, either with another neuron or with a muscle fiber or with a gland. When we talk about talking about a neuron communicating with another neuron, we call the neuron doing the communication, right? The neuron that is whose axon, whose synaptic terminal is synapsing with the dendrites of another neuron. We call that neuron the presynaptic neuron. And then we call the neuron that is receiving the signal via its dendrites the postsynaptic neuron. So, and we'll touch on this again here in a moment, and I said this yesterday, but just as a reminder, when we talk about communication of one neuron to another neuron, many times you have your postsynaptic neuron being stimulated by more than one synaptic knob from more than one neuron. So that becomes really important when it comes to the summative effect that we see of excitatory and inhibitory postsynaptic potentials, and having them add up to or having those potentials be able to reach the threshold value that's required for an action potential to fire. That said, we also do have what are called electrical synapses. So with electrical synapses, the pre and the postsynaptic neuron are actually physically connected to one another. And what occurs during an electrical synapse is you have the action potential in the presynaptic neuron reaching the synaptic knob, and then that action potential is transferred to the postsynaptic neuron through gap junctions, which are kind of like little pores that are present in the membrane of the cell. Electrical synapses are very, very, very fast. They're not incredibly common. You find them in, for example, in specific areas of the brain. You find electrical synapses in the eyes. And the reason that they're not, or I guess we speculate the reason that they're not as common as chemical synapses is that there's less room for kind of fine-tuning the activity, right? Again, action potentials are all or nothing, but reaching the threshold value that allows for a cell to fire off an action potential, that is not all or nothing, right? Again, we'll talk about that more when we get to the excitatory and inhibitory postsynaptic potentials. So with an electrical synapse, it's something that kind of can't be adjusted as finely as what can occur when it comes to communication using a chemical synapse or neurotransmitters. Okay, so now we're going to go through and we're going to talk about the different neuroglial cells that we have both in the central nervous system and in the peripheral nervous system. So again, glial cells are not electrically excitable cells. They do not generate and propagate action potentials, but what they do is support, nourish, protect neurons and enable them to do their jobs, which is to generate and fire action potentials, to do that more efficiently. So we find glial cells associated with both the central and the peripheral nervous system. Glial cells, unlike neurons, are highly mitotic. So we talked about uh, amitosis and longevity being characteristics of neurons, but for glial cells, these cells have a much shorter lifespan, if you will, and again are highly mitotic, so they proliferate rapidly. Um, so what we're going to do, or what I'm going to do now, is go through the glial cells that we have for the central and for the peripheral nervous system. And I expect you to be able to tell me what these glial cells do functionally. And I expect you to be able to tell me on an exam whether or not they are associated with the central or the peripheral nervous system. The spoiler alert, the vast majority of glial cells are found within this. We have more glial cells associated with the central nervous system than we do with the peripheral nervous system. So the first of the central nervous system glial cells we'll discuss are the astrocytes. Astrocytes are called astrocytes because histologically speaking, they look like stars, right? And they look like stars because they have these long appendages that come out, the pedicels that come out, and what they do is you find astrocytes wrapping these pedicels, these appendages wrap around blood vessels that are associated with the central nervous system. And as such, they form what's called the blood brain barrier, which is a highly selective barrier that lets very few things in and out of the central nervous system. Small nonpolar entities are able to cross the blood brain barrier, but it's an incredibly selective barrier. That's what these perivascular feet is another name for the pedicels that I was just discussing. Um, Astrocytes also play a really important role in essentially forming what you can think of as the scar tissue for the central nervous system. So, if, for example, somebody sustains head trauma and the neurons in the brain are damaged and that tissue is destroyed, the brain doesn't, because the way that neurons communicate with one another, brain tissue doesn't allow for holes, right? Or doesn't deal well with holes. If there's a hole, that essentially makes it so that synapses can't occur, right? Because they need something to occur across. And so astrocytes are kind of will fill in gaps. If neurons die in the brain, if that tissue is destroyed, neurons, I'm not sorry, not neurons, astrocytes will fill in that gap and form kind of, like I said, you can think of it as brain scar tissue. Um, and that is helpful in many ways. It's certainly better than having a hole in that tissue. However, again, astrocytes are not capable of propagating action potentials, and so they slow down where you have that astrocytic scar tissue within the brain. That is an area that is typically not considered a functional. That tissue is not function. It can't function the way it did before that tissue was destroyed. Astrocytes also play a really important role in during the neuronal development, embryologically speaking. Uh, Astrocytes play a really important role in helping to essentially tell the neurons where to go during development, if you will. They play an important role, that the chemicals that they secrete play a really important role in stimulating one neuron to form synapses with another. Okay, we also have what are called ependymal cells. So when you think of ependymal cells, think of cerebral spinal fluid. So these are the cells, they line what's called the ventricles of the brain, right? Which hopefully you've seen. No, you probably haven't yet because you guys are taking the exam, the lab exam, but you will see when you get to this in lab. Ventricles are spaces within the brain. So within what we call the lateral ventricle, which is the largest ventricle that we have in the brain, the lateral ventricle is lined with ependymal cells. Um, and ependymal cells, along with associated capillaries, form what we call the choroid plexus. And so this is the structure that secretes cerebral spinal fluid. So the vast majority of cerebral spinal fluid is generated within the lateral ventricle, and then it flows throughout the brain in the subarachnoid space. Okay, we also have what are called microglial cells. So think of microglial cells as being the immune cells of the central nervous system. These are phagocytic cells, much like macrophages and monocytes. They go along and they gobble up cellular debris, and are able to recognize and protect the central nervous system to some degree against pathogenic foreign entities, so foreign cells or cells that are displaying foreign antigen. For what it's worth, when we look at the glial cells within the central nervous system, microglial cells make up the smallest percentage. Um, and again, they're incredibly important at helping to prevent infection of the central nervous system. That said, if they are not able to control infection within the central nervous system, that can become particularly problematic because of the blood-brain barrier. It's very difficult to deliver medications to the central nervous system because that's such a highly selective barrier.
Are microglial cells connected to T cells?
Microglial, well, in the sense that they're both immune cells, they are, um, but they're very different in the way that so T cells and B cells are a part of what we call the adaptive immune response, and they're highly antigen specific. So they recognize each receptor for T or B cell recognizes one and only one type of antigen. And microglial cells would be more like macrophages, so which recognize debris, are able to recognize debris, and they're able to recognize what we call conserved regions of various pathogens, which are so they're not antigen specific, but they're able to recognize something as being. I kind of hesitate to use the word foreign because people think of that in the context of specific antigen, but I kind of suffice it to say they're able to recognize something as debris or as being an infected cell or these conserved regions of pathogens and gobble those entities up. T cells and B cells don't actually undergo phagocytosis, which is a type of endocytosis in doing their job. That was a long-winded way of saying kinda. Yeah, they're both immune cells.
Okay, thank you.
Yeah, no problem. Um, so oligodendrocytes. So we're going to talk here in just a moment about myelination. So oligodendrocytes are the cells that myelinate the axons of neurons in the central nervous system. They do this in a very different way than what we call Schwann cells, which are the cells that myelinate axons in the peripheral nervous system. So just a little bit of background, excuse me, on myelination. Myelination is, you can think of it, so if you're thinking of the action potential, as you should, as an electrical signal, right? And you can kind of think of the axon of a neuron as being like a wire, right? And you can think of the myelination as being the insulation of the wire. So the more myelinated an axon is, the faster the action potential, the faster the rate at which the action potential will be propagated along the length of that axon. Okay, myelin itself it's made up of a big percentage of adipose tissue, so it's a fatty insulative sheath that goes around the axon. And again, what it does is it increases the rate at which the action potential is propagated along the length of the axon. The way oligodendrocytes myelinate axons within the central nervous system, you can see it in this picture. So this would be one oligodendrocyte right here. And you can see it has several appendages. And each one of those appendages comes off of the cell body of the oligodendrocyte, and then it wraps around a portion of an axon, right, for a neuron that it's adjacent to. So for one oligodendrocyte, it can have appendages that can myelinate, a handful of segments of axons from neighboring neurons, right? This is again very different than the way a Schwann cell myelinates within the peripheral nervous system, because a Schwann cell, each individual Schwann cell will wrap around just one small segment of an axon in the peripheral nervous system. And I'll show you a picture of that in a moment that I think will help kind of drive that point home. The other thing to take note of between the myelination, so where the different portions of the axon where they're myelinated, where they come together, there's a little space. It's called the neurofibril node. When we talk about the Schwann cells in the peripheral nervous system, we call these the nodes of Rambier. So this node is when an action potential is being propagated along the length of an axon. Essentially, it only has to be generated where those nodes exist. Okay, so the myelination, it allows for, the action potential, the gates are, sodium gates are opening, sodium's rushing in, you get, the value going from -70 up to -55, which is the threshold value for a neuron. And then that opens up the potassium gates, right? This is the depolarization repolarization event, which is what the action potential is. All of that has to occur literally physically by opening and closing voltage gated sodium and potassium channels, right? So that occurs when you have myelination. That process only has to occur where these little nodes exist, right? And so essentially what happens is the action potential jumps from node to node, because the ions can just diffuse across where the myelin exists. We call that kind of conduction saltatory conduction, and it's very, very, very fast. And it's different than if you didn't have any myelination at all. You'd have to generate, again, that the action potential would have to, it's due to the opening and closing of the voltage gated ion channels. So that would have to occur all the way along the length of the axon. And so that would take a lot longer than it does if it just has to occur at those nodes. Okay, which kind of nicely brings us to this slide. So that's it for central nervous system glial cells. So within the peripheral nervous system, let me start first with the Schwann cells, which are also called neural lemmacytes. So Schwann cells are the functional equivalent of oligodendrocytes for the peripheral nervous system. And so what they do is they myelinate axons in the peripheral nervous system. Again, as I mentioned, they do this in a very different way than the oligodendrocytes. So what they do is, you can see here, this is showing you a multipolar neuron, right, in the peripheral nervous system, the axons in the peripheral nervous system. And this Schwann cell, and I kind of default for what it's worth to just calling these cells Schwann cells instead of neurolemmacyte, because that was the more commonly accepted term when I was learning this material. But both terms are acceptable to describe these cells. So these cells, these Schwann cells, wrap themselves, each cell wraps itself over and over again, kind of like like a jelly roll, around one segment of an axon. So what you're seeing here in purple is the nucleus for this particular Schwann cell that's wrapping around the axon, right? So you have, and so in doing this, I guess the point is, in doing this, you get a thicker layer of myelin around the axons of the peripheral nervous system. And you also have, if a portion of this axon gets damaged, you have because this is made up of individual cells, it's easier for the glial cells to be prompted to go and undergo mitosis and then to remyelinate this segment of the axon. So it's having the way in which Schwann cells myelinate, kind of the take home is that the way in which Schwann cells myelinate axons in the peripheral nervous system is a more effective way of myelinating than it is within the central nervous system, which makes sense largely if you just think about the fact that within the peripheral nervous system, you'll have a motor neuron that can have a very, very long axon that goes down and innervates, some structure in the periphery, your palm, for example, right, or somewhere on your leg. So these cells, these axons, have to span quite a bit of just lengthwise. They have to cover quite a bit of area. Within the central nervous system, all of that tissue is packed either within the cranium, right? That's where the brain is located, or in the spinal cord is packed within the vertebral canal. So you have a much more kind of densely packed, the cells that make up that tissue are much more densely packed. So having extensive myelination is just not as, it's not as necessary as it is in the peripheral nervous system, because the action potential in part just has a shorter distance to travel. As I mentioned, the spaces between the Schwann cells are what we call the nodes of Ranvier, or the neurofibril nodes. The kind of conduction that occurs when you have a myelinated axon is called saltatory conduction, and it's the fastest type of conduction that we have. And it's because you get generation of the action potential only in where the nodes are located, and then where the myelin is, the ions just diffuse through, instantaneously for all intents and purposes. So you only have to generate the action potential in a few locations. So it's very, very, very fast. Okay, so when it comes to cancer that's associated with the nervous tissue. This is what I want you to know. So, which you could kind of, I think, deduce based on your knowledge of neurons versus glial cells, right? The vast majority of brain cancers or brain tumors, and I guess I should say, let me back up a moment. Tumors don't in the brain can be what we call benign, right? So they're not technically mutated. They're not cancer cells in the sense that they won't break off of the tumor, get into circulation and travel to a distant location to set up shop, so to speak. So benign tumors don't that process is called metastasizing. So benign tumors don't metastasize. That said, benign tumors can be very destructive and problematic when they are in the brain because the brain is in an enclosed cavity, right? And again, you have tissue that's packed into a relatively small space. And so oftentimes, even benign tumors can be inoperable and can be put pressure on tissue and therefore which will therefore result in death of a tissue adjacent to the tumor and cause quite a bit of destruction, right? So that's kind of the main difference between the benign and malignant tumors is that the malignant tumors have the ability to metastasize and spread elsewhere in the body, and benign tumors do not. But both can be very problematic. So going back, however, to the types of cells that we find that these tumors originate from, the vast majority of tumors, benign or malignant, derive from glial cells, right? Which makes sense because glial cells are highly mitotic. Very few cancers of the nervous system are of neuron origin. And that's just because neurons are mostly amitotic. That's not to say they don't exist. They certainly do. Most of them happen to be cancers that started back during embryological development and fetal development, and then oftentimes are not detected until sometime in childhood, and then maybe even into adulthood. I'm not 100% sure, but all the same, most cancers are what they call gliomas. So gliomas are tumors of glial origin.
Um, I think you may have skipped over the satellite cells like two
Oh,
slides ago.
I sure did, Nathan. Thank you.
Thanks.
Right. See, that's what happens when I try and be fun and go out of order. Okay. Satellite cells. Let's go back to this peripheral nervous system cell. And luckily, I don't have too much to say about them. So, satellite cells are cells that wrap the cell body of neurons in the peripheral nervous system. And we don't entirely understand what they do. We know that they are important in protecting and supporting and nourishing peripheral nervous system neurons. And they uh, help to eliminate waste that's produced by those neurons and regulate nutrient exchange. But as I said, um, that's kind of yeah, that's all I have to say about them because they're still kind of their full function is still something that's an area of relatively hot research. Okay, so let me see what is on the slide that I haven't already said. So again, I think I've said I don't want to be too redundant, but myelination, think of it as the insulation of a wire, increases the rate of propagation of an action potential. Uh, Myelination is accomplished by Schwann cells, which are called neurolemmacytes within the peripheral nervous system, and oligodendrocytes in the central nervous system. Um, and then the only other thing, I think that's actually it. That's all I have for this slide, you guys, because I already talked about it earlier. Okay, so with unmyelinated axons, of course, we are not going to have saltatory conduction that takes place in unmyelinated axons. We instead call this type of conduction continuous conduction. Um, uh, the action potentials propagated by neurons with unmyelinated axons is much slower. Um, but it's worth mentioning that even for unmyelinated axons, within the central nervous system, or I'm sorry, within the peripheral nervous system, um, and kind of to some degree with both, but within the peripheral nervous system, these cells are still associated with myelin to some degree. And what I mean by that is that you still have portions. So for example, you have a cluster of axons that are put together, which we know we call a fascicle, but this is not an entire fascicle. This would be a smaller cluster than that, but they kind of get sandwiched. They're oftentimes somewhat protected by a myelin sheath from a nearby Schwann cell, for example. So again, the myelin sheath doesn't wrap around them, but the myelin sheath plays a role in kind of holding the axons that are not myelinated, if that makes sense. But it doesn't increase the rate at which the action potential is propagated because it's not insulating the entire length of the axon. And so the action potential still has to be propagated along the entire length. All right, so for clinical view, one example of a clinical view that is associated with oligodendrocytes is what we call multiple sclerosis. This is an autoimmune disease. And in this autoimmune disease, the person's body starts to see the oligodendrocytes as being foreign and attacking those cells. And what happens is you get this progressive demyelination of axons within the central nervous system. And in addition to that, you end up with scar tissue formation because of the inflammation, because of the immune response that's occurring. We know as far as under the risk factors associated with multiple sclerosis, we know that there tends to be a hereditary component. Also damage, if there tends to be a breach of the blood-brain barrier, that is one suspected way that multiple sclerosis might be initiated. But what happens over time, because it's progressive, is that individuals with this autoimmune disease start to lose control of a variety of functions that essentially affect every system of the body. So visual impairment, musculoskeletal problems, again, every system for the most part is impacted when it comes to multiple sclerosis, because again, the myelination is disrupted. And so the action potential is not firing at the rate that's required to sustain normal activity for that organ that's associated with a particular system. Another disorder that is associated with homeostatic imbalances of myelination is something called Guillain-Barré. So here's what I'll say about Guillain-Barré. Guillain-Barré is one of those, it's a syndrome, right? So what characterizes this syndrome is a loss, this kind of onset of paralysis that can be either be very, it can be limited to one very specific area, like maybe the left side of the face, but can, in some cases, spread down to the diaphragm. And if you end up with diaphragmatic paralysis, that would kill you if you weren't intubated, because you wouldn't be able to respirate. Guillain-Barré, we don't know. It's not an autoimmune disease, but we know that it is associated with inflammation, and it is specifically a type of inflammation that causes loss of myelination in the peripheral nervous system, so peripheral nerves. And so oftentimes people detect Guillain-Barré because their fingers will start to tingle. And then again, that sometimes it spreads. It can spread up to the face. It can spread to the chest. It tends to be self-limiting. We don't really, there's not one specific infectious agent that has been identified as being the causative agent of Guillain-Barré. We do know that this syndrome is something that you see after viral infections in a lot of individuals. So we know that it's associated with an inflammatory response. This is also for people, I know that as everybody should, there's a lot in the news right now about vaccine and vaccine development. And one thing that gets continually brought up, specifically, I would say by the anti-vaxxer groups, is Guillain-Barré. And it's because, to be fair, Guillain-Barré can be something that if somebody gets vaccinated, it can be a side effect, if you will. It is very rare, but for some people, that can happen. It is more common, for what it's worth, we see Guillain-Barré more often in individuals who get an acute viral infection, right? But the way in which vaccines work is they initiate an immune response. So for some individuals who are susceptible, a vaccine, of course, could trigger it. But again, so could a viral infection. So for the most part, people recover really well from Guillain-Barré. That said, if it causes diaphragmatic paralysis, and you're not in an area where you can get to an emergency room and get intubated, then of course it could result in death. Okay, with axon regeneration, here's what I want to say about axon regeneration. So when it comes to the central nervous system axons do not regenerate very well. And it is much, much, much more difficult to stimulate axon regeneration within the central nervous system. It's not impossible, but it's much more difficult. Within the peripheral nervous system, if you think about I use the example of a neuron that is innervating, the palm of your hand. Peripheral nervous system axons are much more likely to be damaged, right? If you had a laceration on the palm of your hand, for example, right? So peripheral nervous system axons are much better at regenerating as long as the cell body of the peripheral nervous system neuron is intact, and as long as some of the neural lemma remains, then regeneration is and it happens all the time, right? We get cuts, we get crushing injuries. Again, as long as for the most part, as long as you have that cell body intact, you will likely get successful regeneration. Regeneration is clearly more likely if there's less damage, and it's also more likely if the damage is closer to or if the damage that occurs is closer to the site that is being innervated. So the closer, basically another way to say that is that the closer the damage is to the cell body, the worse the damage. And that's just because anytime you damage the cell body, it makes it, and again, neurons are mostly amitotic, it becomes difficult or not possible at all for that neuron to regenerate the axon that was damaged. Okay. So here are the basic steps that occur during regeneration. So, and this is a picture that comes from your textbook. So, for example, if the axon gets severed because of a laceration, the proximal portion of the axon will swell and kind of seal off. And then the distal portion, which would be down here, starts to disintegrate, right? And that's called Wallerian degeneration. However, the neural lemma, as long as there's some neural lemma that remains, that neural lemma stays intact. And then what happens is you get the formation of what we call the regeneration tube. And then through the regeneration tube, you get essentially the regeneration tube will form around the remaining neural lemma and the portion of the axon that wasn't damaged. And then you just get essentially a regrowth of the axon through the regeneration tube. And then you get the neural lemma that is reestablished in the area that's grown. You can see this swollen portion, this kind of knobby area right here that's growing out. This happens under the influence of various growth factors that are released by the Schwann cells. And something else, one other thing I wanted to say, because I brought up that axon regeneration happens all the time in the peripheral nervous system, but it's very limited within the central nervous system. Another reason why we do not see axon regeneration happening very well within the central nervous system is that not only do the neurons and the oligodendrocytes not secrete growth factors. So I was just saying the Schwann cells secrete growth factors, right? When the axon is damaged. Oligodendrocytes do not do that in the central nervous system. Not only do they not do it, but in fact, glial cells in the central nervous system secrete inhibitory types of signals. And that's really important because, again, the central nervous system, that tissue is located in bony cavities. And if you had an overgrowth of tissue in a bony cavity, there's nowhere for that tissue to go. And so you would start to put pressure on that tissue and that tissue would start to die. And so the axons, the neurons and the axons of those neurons in the central nervous system, again, they both don't have the growth factors to prompt regeneration of the axon. And in fact, if anything, they're told to stay quiescent so that they don't, I guess, outgrow their space, so to speak. Oh, well, here you go. I didn't even need to be that verbose because here that is right here. So again, that's kind of the take homes. Central nervous system axon regeneration is highly limited for the reasons I just discussed. Okay, so now we're going to move on and we're going to start talking about what we call the ultrastructure of neurons. And ultrastructure means what are the pumps and the types of channels that we find along the length of the neuron, and what role do those play in the initiation and propagation of an action potential, and then what does it look like when the action potential gets to the synaptic knob and releases its neurotransmitter, right? And a lot of these topics we've already discussed. So I'm hoping as we go through this, that this is starting to feel very familiar to people, right? I said this yesterday, but just by way of reminder, this material lends itself really well to drawing pictures when you're being tested on this in an exam. I highly recommend drawing little pictures. So if as I'm going through this and you have a question, feel free. I can't see, I don't have the gallery up, but feel free to unmute yourself and ask me a question, because like I said, this should hopefully be familiar stomping ground at this point. Okay, so with all of that said, pumps and channels, right? We've talked extensively about pumps, but just some background info by way of reminder. So pumps we find embedded in the plasma membrane. They play an absolutely critical role in establishing the chemical gradients for the ions that are involved in the initiation and propagation of an action potential, right? We've already talked about sodium and potassium, and we've talked about calcium, but we're also going to throw chloride into this mix and talk about the role that plays in action potential initiation and propagation and action potential. But the pumps are there to establish the concentration gradients. And then what the channels do is they allow, when they are open, for the movement of the ion they are designed to let through. And that ion is going to move down its concentration gradient. So from an area of high concentration for that ion to an area of low concentration for that ion. Another reminder when it comes to channels is that we have two types of channels that we've been discussing, right? We've been discussing chemically gated channels. So chemically gated channels open when a specific neurotransmitter or chemical, that's why they're called chemically gated, binds to that chemical's receptor that is a part of that channel. So the binding of the chemical to its receptor opens the channel and allows the ion that channel is designed to let through, allows that ion to move down its concentration gradient. Voltage gated channels open what opens them is a change in the voltage, right? And that's what an action potential is. It's a change in the voltage of the resting membrane potential, which for neurons is -70 millivolts. So when an action potential, once you get to the threshold value, which for neurons is -55, once you get to that value and the action potential fires off because it's all or nothing, that depolarization event opens up the voltage gated sodium channels, and then the repolarization, or the value that is reached at the kind of height of the depolarization event, closes the sodium gated channels, and the potassium gated channels that are open now are responsible for the repolarization of that cell. Just kind of letting you know what's coming. We're going to discuss the action potential in a lot of detail again. So I've been giving you guys, we've been talking about it, starting really in chapter 10. And so if this feels kind of repetitive, that's what I'm looking for. I'm looking for this to be familiar information for people at this point. Okay, so that's what this slide goes over right here. So for all gated channels, let me back up a minute. For all of the voltage gated and the chemically gated channels, those channels, their resting state is to be closed, right? We also have another type of channel that are what we call leak channels. So leak channels are channels that are always open, and they allow the ion that they are designed to let through to leak down that ion's concentration gradient, okay? And I mentioned when we were in chapter 10 that leak channels play a really important role, specifically potassium leak channels play a really important role in establishing the very negative values that we see for the resting membrane potentials of the electrically active cells such as muscle fibers and neurons, okay? And then again, those pumps are what establish the concentration gradient. So for sodium gated channels, for voltage sodium gated channels, these channels are, because I just said a moment ago that for the voltage gated and the chemically gated channels, that their resting state is to be closed. And so they're either open or they're closed, right? So for the voltage gated sodium channels, there's kind of a little nuance here. These channels, voltage gated sodium channels, have two gates. They have what are called activation gates and they have what are called inactivation gates, right? And this is what they look like. So the inactivation gates look like these little cusps on the inside of the channel, right? And then the activation gate looks like what you would kind of picture in your mind as a gate, a little flap, if you will, that when that flap is closed and covering the channel, this prevents the movement of sodium. And when it's open, it allows for the movement of sodium. Likewise, when the inactivation gate is closed, it prevents the sodium from moving. And when it's open, sodium can move, right? And here's the take home with this. In order for sodium to move across a voltage gated sodium channel, both the inactivation gate and the activation gate have to be open. Okay, that's what we call the activation state. All of the other states of the sodium channel prevent the movement of sodium. And the reason, the speculated reason why we have these two different gates, at least in part, is to help to tightly control the movement of sodium, because sodium is so important. Sodium is what causes the depolarization event, right? It's ultimately what causes the action potential. If an action potential is going to occur, sodium is what's going to bring the threshold value from -70 to -55. So it's so the movement of sodium is very tightly controlled. Okay, so during the resting state of the voltage gated sodium channel, as you can see in this picture, the inactivation gate is open. Where's my cursor? Is open. The activation gate is closed, and therefore sodium can't move, right? In the activation state, both are open, so sodium can move down its concentration gradient. And then in the inactivation state, what happens is that the activation gate remains open, but the inactivation gate closes temporarily, so sodium cannot move through the channel. And then what happens is the activation gate, which again, think of it as like an actual gate, the way you kind of picture a gate, it will finally close, and the inactivation gate will open. However, still sodium can't move. Okay, the only time sodium can move across the channel is when that channel is in the activation state. Okay, so now we're going to go through and we're going to talk about the microanatomy of the neurons, and we're going to talk about where we find the pumps and the channels. Okay, and we're going to identify the different regions. I'm actually going to come right back to this slide, but I want to show you guys this one. We're going to identify the different regions and of the neuron, and we're going to call them one area, or the first area, if you will, the receptive segment. You can think of the receptive segment as being made up of the cell body and the dendrites. Then we have what we call the initial segment. You can think of the initial segment as being the axon hillock. We have the conductive segment. You can think of the conductive segment as being the axon. And then we have the transmissive segment, and you can think of the transmissive segment as being the synaptic knobs or synaptic terminals or axon boutons or whatever terminology. All those things are the same thing. Okay, but let me go back here first. So along the entire length of the neuron, we are going to find both sodium and potassium leak channels, right? Potassium leak channels are found in greater abundance compared to sodium leak channels. And that's again because they play, and that's how they play such an important role in establishing that very negative resting membrane potential of -70, is that there are more of them. And so sodium is constantly leaking out to a greater extent I'm sorry, potassium is constantly leaking out to a greater extent than sodium leaking in. So, and potassium has a positive charge, so it leaves, it's leaking out, and then it drops that resting membrane potential down to -70. Also found along the length of the neuron, we're going to find sodium and potassium pumps. These pumps are pumping 3 sodium out into the extracellular space. For every 2 potassium, they pump in to the internal portion of the neuron, right? And in doing so, they establish that chemical concentration gradient by which we have much more sodium on the outside of the cell than we do on the in, and much more potassium on the inside of the cell than we do on the out. Okay. And then, of course, the pumps also play a role in helping to maintain that resting membrane potential of -70 millivolts, because they're pumping more of a positive charge, the sodium, out than they are pumping a positive charge, the potassium, in. But again, it's
So,
really, oh, yes.
do sodium potassium pumps go with or against the concentration gradient?
So they're pumping against the concentration gradient. So they're pumping into an area of high concentration for that ion. That's a good question. Okay, so this is back to this slide, right? So again, think of the receptive segment as being made up of the cell body and the dendrites. So now we have to talk about what we find here. We already know, of course, that we're going to find those leak channels we just discussed in the pumps that are there to maintain the chemical, to establish and maintain the chemical concentration gradient. But what else do we find here? So within the dendrites, what we find are chemically gated ion channels, right? Specifically, we find chemically gated potassium channels, we find chemically gated chloride channels, and we find chemically gated what we just call cation channels, right? So, and those cation channels are going to allow for the movement of sodium. So these, and really you should hone in here on the fact that these are chemically gated, right? So this tells you that for these channels to open, a chemical has to bind. And that chemical, right? I mentioned yesterday that dendrites are the receiving portion of the neuron. When you have a pre-synaptic neuron synapsing with a postsynaptic neuron, that presynaptic neuron, it's going to be the synaptic knob of that presynaptic neuron, releasing neurotransmitter into the synaptic cleft that's hovering right above the dendrites of the postsynaptic neuron, right? And on the dendrites, that's where you're going to find all of these chemically gated ion channels. So, and another important point, and I'll kind of hammer this in more detail going forward, but the neurotransmitter that is present is going to determine the type of ion that, or the type of channel that opens, and therefore the ion that moves, okay? The other thing I would do when you're taking a test, for example, if you draw a little picture like this to remind yourself of where we find these different structures, make sure and draw in the chemical concentration gradients for the ions that are important in this process, right? So what you would draw is you would draw a bunch of sodium out here, maybe put one in the middle to remind yourself, of course, that there is some sodium on the inside of the cell. You would put a bunch of K plus, K plus, K plus inside the cell to remind yourself that there is a high concentration gradient, or there's a high concentration of potassium inside the cell, and put a couple Ks out here to remind you, of course, that potassium is outside the cell. And then for chloride, so there is a much greater chloride mirrors sodium. So you have much more chloride outside here than you do inside the cell. And the same goes for calcium, which we've not gotten to yet, but we will here. There's a much higher concentration for calcium outside the cell as opposed to inside the cell. Okay. And this becomes these channels are going to become really important when we talk about the effect that neurotransmitters have on binding to their receptors that are a part of these channels and opening them, and what effect that has on the resting membrane potential of this neuron, right? We can have, if you, for example, were to open up, if you had a neurotransmitter that opened up the chemically gated cation channels that are going to allow sodium to pass, that's going to, of course, based on sodium's concentration gradient, sodium is going to rush into the cell and that will start to depolarize the membrane. But what if you have a neurotransmitter that opens up a chloride channel or opens up potassium channels? Well, based on the chemical concentration gradients for these, you're going to have chloride. In that case, if you're opening a chloride channel, chloride will enter the cell. Chloride has a negative charge associated with it. So that will what we call hyperpolarize the membrane. It'll make the value, the resting membrane potential of this neuron, it'll make it lower than -70. It'll bring it even farther away from the threshold value. So it'll make it harder, if you will, to generate an action potential. Same thing with opening up potassium channels. If you open up potassium channels, potassium will leave the cell, and that is going to make the value, the potential value measured here in the cell, more negative. So harder, it'll take longer, or it'll be harder, if you will. Think of it as kind of harder to reach the threshold value that's required to fire off an action potential. Okay. So again, chemically gated channels you find in, let me see how I am on time. Ooh. Okay. In the receptive segment. Then in the initial segment where the axon hillock, I said yesterday that I want you to think of the axon hillock as the trigger zone for the neuron, right? And that's because this is the area, if the threshold value is reached, that value will be reached here in the receptive segment, and it will essentially be passed to the initial segment, and it'll open up the voltage gated sodium channels. If again we get to that threshold value, it'll open up the voltage gated sodium channels here in the initial segment, and then that's where the action potential is fired from, right? It's if you start opening these channels here, this is where the action potential it will be fired from, and it'll pass along the length of the axon. What we find, structurally speaking, in the initial segment and in the conductive segment are the voltage gated sodium and potassium channels that are involved in the generation and propagation of that action potential, right? Those are the events that we have already talked about, where again you reach the threshold, which for a neuron is -55 millivolts. You open up that opens up the voltage gated sodium channels. Sodium rushes into the cell and it brings that value from -55 up in the positive range. I don't care that you know the actual value, but positive 30 more or less. And then what happens at that value is that you have the closure of the voltage gated sodium channels. The voltage gated potassium channels have actually started opening up during the depolarization event, but as soon as you close those voltage gated sodium channels, now you only have the voltage gated potassium channels open. And so at that point, potassium will leave and you drop the value back down toward the resting membrane potential. And we will cover that in a lot of detail in a slide. I just want to say it because that's the process we're discussing right here. And then finally down here in what we call the transmissive segment, which again is made up of the synaptic knobs, what we find here are calcium pumps. And this is right, we've talked about this in pretty good detail when we were discussing the neuromuscular junction. But just as a reminder, what we find here are potassium pumps that pump a bunch of, I'm sorry, did I say potassium? Calcium. Jeez. Calcium pumps that pump a bunch of calcium out of the synaptic knob, and they establish a concentration gradient for calcium by which there's a lot of calcium outside and not a lot in. And then we find voltage gated calcium channels in this area. So if and when an action potential is generated here at the axon hillock, it'll spread along the length of the axon. It'll reach the synaptic knobs or the synaptic bulbs or synaptic terminals. It's a change in voltage, so it'll open up the calcium channels. Calcium will rush into the synaptic knob, and then it'll coat the vesicles, which are coated in a negative charge, those vesicles holding the neurotransmitter, and ultimately allow those vesicles, because they're now coated with a positive charge, because calcium is positively charged, allow those vesicles to merge with the plasma membrane, undergo exocytosis, and dump their neurotransmitter into the synaptic cleft, which then will allow that neurotransmitter, if there are receptors for it, to bind to the chemically gated ion channels and the receptive segment of the postsynaptic neuron. Whew, okay. Oh my gosh, let's stop there. We'll stop there. We only have, so what we'll do on Monday, there are only 20 slides that make up the rest of the chapter 12 material. So we'll finish those last 20 slides next week, next Monday, or actually on Sunday, I'll open up, well, this is week 10. I'll open up week 11's folder. And so you'll have in there, I'm not going to dump chapter 12 part 2 into week 11. I'm going to leave it in week 10, which is where it is right now, but we'll finish that PowerPoint and then we will go on and you'll see the brain PowerPoints in the week 11 folder. And so we'll start in on part 1 of the brain. Okay, so I'm